Okay, this is the second recording to finish up lecture 7. And this part is just going to focus a little bit more on the exact project, uh, how the approach is being made to contribute to changes in vein density. So it's not as trivial as it may seem, and I think this is something that a lot of people don't know, which is that just because we have full genome sequences for some organisms, having a DNA sequence doesn't tell us everything. It may tell us what protein is being expressed, and maybe we can, figure out some more things, but we still need to do a lot of work to understand exactly how different genes are having different roles. So there's a few things, sort of unknowns, and we're going to be dealing with a large number of genes because we don't know yet which ones might be the most important. So we're not aware yet exactly which genes contribute to vein formation and how they're spaced and so on. One thing we do know, though, that among all plants, oxin, it's a plant hormone. Those of you who took 333 are already a little bit familiar with oxin because I talked about it in at least a couple of the lectures. It's a plant hormone that is involved in many developmental processes. It is involved in forming where new leaves are going to form, different aspects of sort of the polarity of structures and so on. And it's kind of the most important hormone, and it does a lot of different things in the plant. And one of the features of auxin signaling, this little hormone, is that it gets transported directionally. So during different stages in development, it's being sent towards the shoot, and then it's being sent down towards the root, and so on. And this is done through a series of transporters, which you don't need to really know about, but just to be aware that sort of directional transport of this hormone is part of how it functions, because it will generate little areas where the auxin accumulates, and then it initiates signals that are required for some process. So this is the most common one, which is indole 3-acetic acid, IAA. There are some slight variants that probably do different things, but this is as much as you need to know in this case. And so this is some work from quite a long time ago. It's determined that if you chemically inhibit the movement of auxin in early plant development, you will get an increased vein density. So this is in seedlings, the first and then the fourth leaves. They've been stained with fluorescent marker. I'm just looking at it. I don't know exactly which marker it is in this case, but basically the fluorescent is the developing veins, and you can see this is control. So these are untreated plants, and this is the treated ones, and this is an inhibitor of auxin transport. If you didn't allow it to move at all, of course you would not have viability, but if you inhibit some of the transport, you can see a real difference in both the number and spacing of those veins, right? There's a lot more of them, and they're crammed together. And so this is just it shows that there's kind of like a threshold. So if you only treat with a mild dose, or you treat with a higher dose, the amount of inhibition that's observed is it's like there's a limit to how much of an effect it can have. But if you look at, if you look at, this is in wheat, the previous one was Arabidopsis, but it shows that there's a statistically significant increase in the number of veins. And I point this out, this one in particular, because Mario was a student in my 302 and 357. And he once asked me if I thought he would be suitable to do research, and he's gone on to have this amazing career. He did a PhD at UBC, and he was studying childhood cancers and things like that. And he's now, I think he's a major scientist in some corporation. It's a U.S. based corporation. But so starting out here at SFU, you can still end up doing some really interesting things. This was one of the. He was super smart, but it's not just being smart. Smart is nothing unless you are also really interested and curious and motivated. And he had all of those things. He was a terrific student. And this is a little bit more of the work. This is actual rice in this case. So they've studied the same process in a number of different plants. And if you have, this is not chemically inhibiting, but actually mutations that have defects in oxin signaling. Now these could be oxin transport, but they could also be the proteins that are required to respond to the oxin. So you might have plenty of the hormone about, but if you don't have the transcription factors that then respond to it, then again, you have a defect in the signaling. And so this particular mutation, they showed that the plant size is smaller. They showed that the amount of seeds are much smaller. But they also showed that there was a difference in the density. So they had actually, that should say fewer. I had to get this from Jim, and he's Swedish, and so his spelling is sometimes more Swedish. But the idea is that the mutant actually ended up having fewer leaf veins. So notice that we see evidence of a role of auxin in regulating leaf vein density, but the roles that we're observing are not the same in different plant organisms. So this is why we need to, continue studying this a lot. Where is the thing? There it is. Okay, so the approach is we're using an organism called Soteria italica. It is called the foxtail millet, and there's just a picture that I should have put the citation underneath. Millet is a crop that you can eat the seeds. They're very tiny, so you have to have lots and lots and lots of it to get a good yield. But it is, some of you probably have eaten millet a lot of times. And we're using basically this plant rather than rice because it's just, there's a couple of reasons. One is it's easier to work with. And so when you're transforming the plants and then you're culturing them and so on, it's easier to do that. It's a close relative of rice, and it also already does C4 photosynthesis. And so therefore, we're working with this organism, but we're going to try to find genes that when we knock out those genes, you get a decreased vein density in the foxtail plants. If that's the case, if you knock them out and there's fewer veins and they're not as, they're spaced wider, then we would expect that the normal role of that gene would be to promote the formation of veins and, to have increased density. So that's the idea. And we're going to identify those in foxtail, but then the idea is to take the equivalent gene in rice and then do knockout of that gene and see whether it has the same or a different role in vein spacing. And so we're using I guess you'd call it a candidate gene approach. We're going to look at a whole bunch of genes that are related to auxin signaling, either the production of auxin, the transport of it, or receiving it, having the receptors for it and the transcription factors that work downstream of it. And so the candidate genes, the genes that we have identified to work with, are ones that are, the ones that are known in corn to have something to do with these processes, and that might be associated with the anatomy that is needed for C4 photosynthesis. And so they might have a role, but we don't know what those roles are necessarily. Okay, so that's kind of where that's where the field is. Okay, there's some genes that are thought to have some role in the performance of C4 photosynthesis, but we don't know which genes have which roles yet. And this is part of the work that's going to be done. So it's something that we may find out from your constructs. Okay, clicker questions. Okay, so the genes that we've targeted, that Jim has targeted, Jim and his students probably, they regulate the synthesis of oxin, right? So they're transcription factors that are involved with generating those enzymes that are required for making oxin, or the transporters. Remember I said there's directional transport. So if you have a plant cell, two plant cells beside each other, there's a transporter that sits at one end of one cell to move the oxin out, and then there's an importer on the cell on the other side. So you have the combination of an export and import to move oxin in a particular direction. And if you want to change the direction of transport, you then have to move the transporters to a different part of the cell to send the oxin in a different direction. There's quite a number of these, and they're pretty well known in how they affect the transport of oxin, but they haven't necessarily been examined in detail for specifically their effect on veins and so on. Receptors that recognize and bind to oxin, and then downstream targets. So these would be, so the receptor will recognize and bind oxin, and then there will be signals to initiate transcription factors that then, in the cell that's received the signal, now it's going to form a root, or now it's going to start forming a flower instead of a leaf, or something like that. And so these are transcription factors that change the gene expression in the cells that are responding to oxin. So each pair of student has been assigned a gene. And as I said, they've been identified in corn, which is a C4 plant. And they are possibly required for the anatomy, and especially ones we're interested in are the ones that will induce the formation of veins. Okay, so that is the end of the intro. On Monday, I'm going to talk about the actual process of how you're going to find the CRISPR oligos and so on, and how to submit them to me for the first part of the report, and then there are some questions to answer for the second part. And all that will be covered on Monday.